Structure of eukaryotic cells. So we're going to start off by looking at an animal cell. We'll look at all the organelles in that, and then we'll add on the extra ones at the end that will also be found in plant cells, fungal cells and algae cells. These are all examples of eukaryotic cells. So just like any cell, eukaryotic or prokaryotic is going to be surrounded by a cell surface membrane. This is a selectively permeable barrier, and we're going to look at this in more detail in another topic. The defining feature of the eukaryotic cell is the nucleus, and you do need to know how to label the different parts of the nucleus. So inside the nucleus are the chromosomes, which are made up of long linear DNA, which is tightly coiled around proteins called histones. This is why the nucleus controls the cell's activities, because these DNA contains genes which codes for proteins. So the DNA, sections of DNA gets copied into messenger RNA through transcription, which will then leave the nucleus to go to the ribosomes. The ribosomes are made in the nucleolus, which is the dark patch. Ribosomes are made of proteins and ribosomal RNA. Outside the nucleus, keeping it all together, is a double membrane called the nuclear envelope. And in order for the messenger RNA and the ribosomes and everything else to get in and out, it has nuclear pores, which are just gaps, which allows all of this stuff to leave. So once we come out of the nucleus, our messenger RNA comes out of the nucleus, we need to actually synthesize the proteins, and the organelles involved in protein synthesis are the ribosomes. They can be found free in the cytoplasm, or they can be bound to a system of membranes called the rough endoplasmic reticulum. So the ribosomes on the surface of this will synthesize the proteins. They'll go straight into the inside of the rough endoplasmic reticulum, where they get folded into their secondary and tertiary structures, and therefore the function of this is to transport the proteins. So it can package them into vesicles, which are just small membrane-bound sacs, which kind of bud off from the rough endoplasmic reticulum, and these can store or transport these protein molecules. So normally next what would happen is these would get transported to the Golgi apparatus, which is where these proteins can get modified. For example, we might want to add carbohydrates to form glycoproteins. Again, just like the rough endoplasmic reticulum, this is then going to package the proteins into vesicles, specifically Golgi vesicles, and these could get transported, for example, to the cell membrane, where they can again fuse with the cell membrane and release their proteins into the extracellular space. This is called exocytosis. A specific type of Golgi vesicle that can be produced is called a lysosome. This contains lysozymes, which are hydrolytic enzymes. These enzymes can be released to break down pathogens, and they can also be released to break down worn-out cell components. So now we have another type of endoplasmic reticulum. This time it doesn't have any ribosomes on the surface, so we call it the smooth endoplasmic reticulum. This one has a role in synthesizing and processing lipids like cholesterol and steroid hormones. We have our really important organelle, the mitochondria. Be really careful here, really specific with how you describe its function. So it's not just the site of respiration, it's the site of aerobic respiration, the part that requires oxygen. And this therefore produces ATP. It doesn't produce energy, it produces ATP, which releases energy. We will have a little look at the ultrastructure of this organelle. We're going to come back to that though, because it's very similar to the chloroplast. So now some organelles that are found in plants, algae and fungal cells. So the cell wall, not found in animal cells, but it is found in plants, algae and fungi. This would be found outside the cell surface membrane. So that's how you know which one's the cell wall. It tends to look thicker and it's outside the cell surface membrane. In plants and algae, it's made of cellulose, which is a carbohydrate, specifically a polysaccharide made of beta-glucose, chains of beta-glucose cross-linked together. And in fungi, it's made of chitin, which is a nitrogen-containing polysaccharide. But in both of these, the cell wall provides mechanical strength and therefore helps the cell from changing shape and bursting when it gets put under pressure due to entry by osmosis. It is permeable, it's not selectively permeable like the cell surface membrane. Stuff can just go in and out of it. Then we also have the vacuole, which is normally very large, and it is permanent in plant cells. It's surrounded by a membrane called a tonoplast, and it contains the cell's sap. Its function is to maintain turgor pressure in the cell and therefore support the plant, and also to store and isolate some unwanted chemicals. The chloroplast, that's the site of photosynthesis. It absorbs light and it produces organic substances such as glucose. So now we're going to have a look at the structure of the mitochondria and the chloroplast, because you do need to know these in some detail, and they are quite similar. So the chloroplast has a double membrane. The fluid found inside is called the stroma, and there is another membrane inside called the thylakoid membrane, which is what contains the chlorophyll. This can stack up in certain places to form the grana, and these grana are linked together by lamellae. It also contains its own small ribosomes and circular DNA, very similar to prokaryotic cells. The mitochondria also has a double membrane, but the inner membrane is folded to form cristae. This time, instead of a stroma, it has a matrix, and again it has small ribosomes and circular DNA. Cells can become adapted for specific functions in complex multicellular organisms. They can become specialised, and it is quite a common way to test your knowledge of cell structure is to give you an example of a cell, give you some information about that cell, and to suggest how its structure relates to its function. So normally it will be that it has more of a certain organelle, which links to the function of that. So say, for example, a cell that makes lots of enzymes or makes lots of antibodies, you'd have to recognise that these are proteins, and therefore it would have more ribosomes, more rough endoplasmic reticulum. So those sorts of questions are really just an opportunity to show the examiner that you have knowledge of that particular organelle, the function of that. Another common one is some sort of cell that needs a lot of energy. For example, it undergoes a lot of active transport, or a muscle cell, it needs the energy to contract, and again that would have lots of mitochondria. So specialised cells can be organised into tissues, which is a group of cells with a similar structure and function. They often have the same origin. Then organs are aggregations of tissues performing specific functions, and an organ system is just a group of organs working together to perform specific functions, and an organism would be formed of many organ systems. The structure of prokaryotic cells and of viruses. So all you really need to know about prokaryotic cells is an overview of their structure, but particularly be able to compare it to eukaryotic cells. So like any cell, we have a cell surface membrane. People sometimes get confused with this. Now, a prokaryotic cell doesn't have any membrane-bound organelles, but it is still surrounded by a cell surface membrane. The defining feature of a prokaryotic cell is that they don't ever have a nucleus, although it still does have DNA. Every living organism needs DNA. So the DNA is just free floating in the cytoplasm, not contained within a nucleus. We can sometimes call it a nucleoid. The DNA is different though to eukaryotic cells. Unlike eukaryotic cells where it's long and linear and associated with proteins, the DNA in here is short and circular and not associated with any proteins. Therefore, we can't call it a chromosome. Because it has DNA, it's still going to have genes, so therefore it still is going to code for proteins. So therefore it's still going to need ribosomes, but in this case the ribosomes are much smaller. We can call them 70S ribosomes. Now, like plants, algae and fungi, it does have a cell wall, but again the structure is different here. Instead of being made of cellulose or chitin, it's made of murin, which is a glycoprotein. Sometimes prokaryotic cells, but not always, might have a capsule around the outside. You can sometimes see this being called a slime capsule. They may also sometimes have flagella, but again, not always. Depends on the species of bacteria and also the conditions in which they're living in. Sometimes they might also have plasmids, which are small circular bits of DNA, and they often contain genes which code for things like antibiotic resistance. So to summarise the differences then, the prokaryotic cells don't have any membrane-bound organelles, for example, no mitochondria, no endoplasmic reticulum. They have no nucleus containing DNA, however their DNA is just floating free in the cytoplasm. The structure of their DNA though is different, so it's circular, whereas in eukaryotic cells it's linear, and also it's not associated with proteins. It's also much shorter. The cell wall, if you're comparing it to an animal cell, then obviously a prokaryotic cell does have a cell wall, whereas an animal cell doesn't. But if you're comparing it to a plant cell or an algal cell or a fungi, then the cell wall is just made of something different. So it's made of murin as opposed to cellulose or chitin. They do both have ribosomes, but the ribosomes have a slightly different structure, different size. So they're small 70S ribosomes in the prokaryotic cell, whereas larger 80S in eukaryotic cells. And they also additionally might have plasmids, might have a flagellum, or they might have a capsule. But again, because they're not always there, I would say that the other options are more of a safe bet, more likely to guarantee the marks. And also, prokaryotic cells are much, much smaller than eukaryotic cells. So now on to viruses. There really isn't much that you need to know about viruses, mainly just the idea that they are acellular and non-living. So viruses are actually just particles. So they don't live, and they're not a cell. They're literally just genetic material, and this could be DNA or it could be RNA. It varies between different viruses, but they could be single-stranded or double-stranded. This is just surrounded by a protein capsid, which is protective. Now, it's quite a common mistake to mix a capsid and a capsule up, but they are different things. And then attached to this, we have some attachment proteins, which is what determines what type of cell it can get into. So these will bind to receptors on the surface of the cell that is going to be its host, and therefore enable the virus to get inside that. Now that's the general structure. All different viruses have slightly different parts. So HIV, for example, we'll look at that in the immunity section. That also has a lipid envelope around the outside of it, which it pretty much steals from its host cell, the T helper cell. Methods of studying cells. So we'll start off by looking at microscopes that use light and microscopes that use electrons. So all microscopes magnify objects. This just means making them bigger. So the definition of magnification is just how much bigger the image of a sample is compared to the real size. Therefore, we can calculate this by using the size of the image and dividing that by the size of the real object. We can also put that in a little triangle where is image on the top and then actual times by magnification, and we can cover which part to decide whether we need to multiply or divide. Just remember when you're calculating magnification that everything needs to be in the same units. Some of the units that people commonly mix up is micrometers, which is the one that looks like a little U. That is just means something times 10 to the power of -6 meters, and that's smaller than millimeters, which is something times 10 to the power of -3. So if in doubt, remember those and just convert everything to meters, or you can try and convert everything to centimeters or everything to millimeters or everything to micrometers, just whatever makes sense based on the situation. So resolution is different. Resolution of a microscope is just the minimum distance that two objects have to be apart, basically to be distinguished as separate. So it shows the level of detail. And often the thing that limits this is the wavelength of radiation used. So for example, light has a longer wavelength than electrons, so it creates a lower resolution. We're going to go through both types of microscopes, briefly mention how they work, but most importantly we'll look at their advantages and limitations. So optical microscopes work by light being focused using a glass lens onto a specimen. The light passes through this, it has to be really thin, and basically regions that absorb more light appear darker. We can use this to see living organisms, however it creates a 2D image. The specimen must be really thin and it's low resolution. That's the most significant disadvantage here, and it's low resolution due to the long wavelength of light. So therefore we can't see small organelles such as ribosomes, and we also can't normally see the internal structures of organelles. It's also got a low magnification. So there are two types of electron microscopes. There are transmission electron microscopes and scanning electron microscopes. Both work by electromagnets focusing a beam of electrons as opposed to a glass lens focusing a beam of light onto a specimen. And then a transmission electron, these electrons are just passed through this specimen. Again, it has to be very thin, and the denser parts absorb more electrons and appear darker. Whereas a scanning electron microscope just does what it says. It scans across the surface of a specimen and it generates a 3D image based on the electrons that are reflected. So both of these are much better in some ways, many ways, than optical microscopes, mainly that they have a higher resolution due to the shorter wavelength of the electrons, which enables us to see the internal structures. So if you're looking at a detailed micrograph of like a mitochondria or a chloroplast that's probably been produced by an electron microscope. They also have a much higher magnification. Now, scanning electron microscope has some additional benefits in that it produces 3D images and it can be used on thick specimens and we can see the outside of them. Both of them do have some significant drawbacks. So our specimens need to be in a vacuum, which means that we can't look at living organisms. And due to the nature of the procedure to prepare it, which is very complex staining method and which takes a long time, we can also get artifacts in it, which are basically things that can look like organelles, but there are things that we actually don't want to be there. So you could be given an electron micrograph and be asked to identify parts of the organelles, like the mitochondria, and this can be quite difficult to do, so it's definitely worth having a look at a range of labelled micrographs of different organelles and just testing yourself on seeing if you can identify them. You'll know when a scanning electron microscope is being used because the image will be 3D. So back to optical microscopes, how to use them to measure the size of an object. This is one of the things that you have to do in the mitosis practical. So on the eyepiece is basically a scale without any units on. This is called an eyepiece graticule, and it's just like a ruler but no units. And it's always there, no matter what the magnification, it will always look the same. But on the stage, we can put like a tiny little ruler and we call that a stage micrometer. And what we want to do is we want to calculate the size of an object, like the size of a cell or something, but we don't want to have that stage micrometer there the whole time. What we want to do is actually at a particular magnification, we want to calculate the size of the divisions on our eyepiece graticule so that we don't have to have the stage micrometer there. We can take it away and then just measure how many divisions on the eyepiece graticule it makes up, and therefore use that to calculate the size of the object. Obviously, the divisions would have different values depending on how much we zoom in. So what we do then to calculate firstly the size of each of the divisions on the eyepiece graticule, we simply just line them up and just calculate the size of the divisions. So we divide one by the other. Then we take the micrometer away and measure how many divisions on the eyepiece graticule that the object is made of. Now, although the size of the divisions, like a ruler, isn't actually on there, there's no units on that, we do know, we would have written down now the size of each of the divisions on the eyepiece graticule. So now to work out the size of the object, we just multiply the number of divisions that it's made up of on the eyepiece graticule by the size of each division at that particular magnification. Now, it's worth pointing out that we would need to recalibrate this at different magnifications. Just going back from there, how do we prepare the temporary mount in the first place? So we use tweezers to place a thin section of our specimen onto a drop of water on a slide. We might then stain it. So for example, iodine dissolved in potassium iodide would stain starch granules in plant cells, which might be found in the chloroplasts. But there are other stains that we can use to stain different parts. So for example, the chromosomes when we want to view mitosis. Then we just carefully lower the cover slip by tilting it and making sure that we avoid any air bubbles. Another way that we can study cells is something called cell fractionation. That just means breaking up cells, separating out the organelles. We might want to study one in particular, for example, looking at respiration in the mitochondria. So we start off, we have to break open the cells. This is called homogenization. We could use a blender or a homogenizer, and that releases the organelles. We then put these straight into a cold, isotonic and buffered solution. So cold because it reduces enzyme activity, so they don't break down any organelles. Isotonic means it has the same water potential as the organelles, which means that no water moves in and out by osmosis, so the organelles don't burst or shrivel and get damaged. And buffers means that the pH is constant, so that the enzymes don't denature. Then we want to filter out all the large debris like whole cells or any cell walls and stuff that we don't want. And then we want to spin it to separate out the organelles. This is called centrifugation. Organelles have different densities, so when you spin them, the heaviest will go to the bottom, and we can basically just keep repeating that. So we start off, we centrifuge it at a low speed. The heaviest organelles will form what's called a pellet at the bottom. So that would start off being the nucleus normally. And then the top part is called a supernatant, and that contains all of the other organelles. Then we need to spin it at a higher speed and again remove the pellet containing the next most dense organelle, remove the supernatant and respin that. We basically just keep repeating this at increasing speeds until we've separated out all of the organelles roughly in order of density. The order of these nuclei will always be the heaviest, and then it will be roughly chloroplast if we're looking at plant cells, mitochondria, lysosomes, endoplasmic reticulum, and then ribosomes will always be the lightest. There will be sometimes some overlap between these, but the nucleus will always be the heaviest and the ribosomes will always be the lightest.